Cynthia Puente, and this is my membrane structure and function concept map. I'm going to start over here talking about the amphipathic molecules. An amphipathic molecule is made up of both a hydrophobic region as well as a hydrophilic region. Hydrophobic means that it is afraid of water, as opposed to hydrophilic, which is a water-loving molecule. Type of amphipathic molecule is a detergent molecule, which is a soapy substance used to solubilize membrane proteins. The hydrophobic tails and hydrophobic heads combined make up the phospholipid bilayer of a cell membrane. The lipid bilayer we are introduced by the sandwich model, which was created by Dawson and Danieli. So it shows us very specific structure of the lipid bilayer. The lipid bilayer makes up the plasma membrane, which surrounds all living eukaryotic cells. And we see the plasma membrane structure within the fluid mosaic model, which was created by Singer and Nicholson. The plasma membrane is selectively permeable, which means that it has a control of what molecules come in and out of the plasma membrane. There are various forces that move molecules across the plasma membrane. One being the electrochemical gradient, which moves anions, positively charged molecules, across the plasma membrane. We also have it by means of osmosis, which move water molecules across the plasma membrane. Another type of transporting force is active transport, which allows molecules to move across the membrane, which is driven by ATP hydrolysis or other means of metabolic energy processes. We have simple diffusion, which moves ions across the membrane as well. Simple diffusion doesn't require the help of membrane proteins, which brings us to the side of the concept map. Membrane proteins are found within the phospholipid bilayer, and there are several types of membrane proteins. We have the transporter proteins, which, as the name states, it transports proteins across the cell membrane. We have your anchor membrane proteins, which anchor proteins to the bilayer, and this allows for cells to interact and identify each other. We have protein attached associated proteins, which attach proteins to other proteins by means of weak non-covalent bonds. We also have receptor membrane proteins, which are able to detect a stimulus that's usually caused by a change in the concentration of a specific molecule. We also have enzymatic membrane proteins, which, as we all should know, act as catalysts to speed up chemical reactions. We have monolayer associated proteins, which anchor the proteins to the bilayer by means of an amphipathic alpha helix protein. And then we also have lipid-linked proteins, which attach proteins to the bilayer with a covalent bond. We also have glycolipids, which are attached to the bilayer. They're attached to the hydrophobic tail region of the bilayer. We also have glycoproteins, which are The proteins are attached to the bilayer. They're covalently linked to oligosaccharide chains. Membrane proteins can also be integral proteins, which are permanently attached within the bilayer. They can also be peripheral proteins that are temporarily attached only to the surface of the bilayer. Another type of membrane proteins are membrane channels. Membrane channels allow for processes like passive transport, which with passive transport you have molecules that move across the cell membrane, but they can only cross by means of these specific membrane channel proteins. Types of membrane channels are just your regular ion channel here. It's a protein or a protein complex. Forms a water-filled channel across the lipid bilayer through which specific inorganic ions can diffuse down the electrochemical gradient. And we have your gated ion channels, which the gates of the protein open up when they are stimulated by a certain stimulus, which includes either a chemical stimulus or either a mechanical stress stimulus. Integral proteins allow for facilitated diffusion. So only molecules can pass through the cell membrane with specific integral proteins. And that is that. Now we're going to switch over to this map. Alright, so here we have two separate maps. We have our types of solution concept map. I'll start off by describing the concentration gradient, which is a gradual change in the concentration of solutes within a solution. So we have our high concentration solutes, which is where the molecules are tightly packed within a solution, as opposed to low concentrated solute, where the molecules are more spread out and there's less of them. So our different types of solutions, we have our hypertonic solution, where the concentration of solutes is a lot greater outside of the cell than inside of the cell. So in human cells or plant cells, when placed in a hypertonic solution, they will shrivel or shrink. We have the opposite, our hypotonic solution, which is when the solute concentration is a lot greater inside of the cell than outside of it. So when the cells are placed in a hypotonic solution, they will swell up until they burst or lyse open. Then we have our neutral solution, the isotonic solution, which is where the concentration of solutes is the same within the cell as well as outside of the cell. So the cells, when placed in this type of solution, will neither shrivel or burst. We'll move over here with our types of molecules. We have our saturated fatty acids, which it's a fat molecule that contains no double bonds between the carbon atoms of the fatty acid chain, as opposed to our unsaturated fatty acids, which contains one or more double bonds between the carbon atoms of the fatty acid chains. We also have our sterols or our steroids, which is a lipophilic molecule. So like the hydrophilic and hydrophobic whereas this is it's a molecule that attached to a lipid or likes lipid molecules. So this will be your things such as your cholesterol that acts as hormones such as estrogen and testosterone. And then here we have our proteoglycan molecule, which is a molecule that is made up of one or more glycosaminoglycan chains attached to a core protein. And that is my concept map for membrane structure and function.